Hello, welcome everybody. This is Victoria Echeverria. I'm a product manager here at CDI. We're just going to wait a couple more minutes to allow people to join in on the webinar, and then we'll get started. Hello, my name is Victoria Echeverria. I am a technical product manager here at Cellular Dynamics International, a Fujifilm company. Thank you very much for joining us today for the webinar on iPS-derived retinal pigment epithelial cells in research and clinical applications. It's my pleasure to introduce our speaker today, Dr. Kapil Bharti, Earl Stadman, tenure track investigator for the Unit on Ocular and Stem Cell Translational Research at the National Eye Institute, National Institutes of Health. Dr. Bharti holds a bachelor's degree in biophysics from the Punjab University in Chandigarh, India, where he graduated with highest honors. This was followed by a master's degree in biotechnology at the Maharaja Sayaji Rao University in Baroda, India, and a diploma in molecular cell biology at the Johann Wolfgang Goethe University at Frankfurt in Germany. Supported by an international PhD student fellowship, he obtained his PhD from the same institution, graduating summa cum laude. Dr. Bharti's research focuses on regeneration of the retinal pigment epithelium. He has also developed 3D retinoid tissue and a 3D ocular tissue chip. He plays a key role in NIH regenerative medicine and is the only NIH intramural investigator supported by the NIH Common Fund for his regenerative medicine projects that aim to start for clinical trials for the use of autologous induced pluripotent stem cell derived retinal pigment epithelium patch for macular degeneration patients. In this webinar, Dr. Kapil Bharti will discuss the use of induced pluripotent stem iPS cell technology to develop in vitro disease models, to study patient-specific disease processes, to set up high-throughput drug screens, and to develop cell-based therapy for retinal degenerative disease. We welcome your questions today, so please use the GoToWebinar interface to submit your questions for our speaker today. So you can just click on that arrow there and submit your questions in the text box. For those on site in Madison, please write your questions on paper and I will collect those. And so we'll save your questions for a question and answer session at the end of the presentation. And without further delay, I will hand things over to Dr. Kapil Bharti.
All right. Can you hear me well?
Yes.
Yes. Okay. Wonderful. And you can see my screen?
Yep.
All right. So we're good to go. Thank you, Victoria, for the kind introduction. As she mentioned that in the next 45 minutes or so, I'll show you kind of the work done in my lab, mainly using human iPS cell-derived retinal pigment epithelium cells, and how we use these cells both for research and clinical applications. So really, this center part is the flow is really the main flow of my lab. We start with patients or healthy donors, somatic cells, reprogram those cells into iPS cells, differentiate those cells, iPS cells into RPE cells, and then use them either in what we call disease in a dish approach, where we make either 2D or 3D disease models and perform drug screens, and with the goal to identify or discover potential drugs that we want to bring back to patients. And I'll show those 2D and 3D models in part 1 and part 2 of my talk. And in the last part of my talk, I'll show you kind of a complementary approach, what we call the cell-based therapy approach, where we're using this RPE cells derived from macular degeneration patients and developing an RPE patch that we hope to bring back to patients as a potential cell therapy. So what are retinal pigment epithelium cells? These cells are part of a homeostatic unit in the back of the eye called the photoreceptors RPE and choroid. The schematic here describes really the kind of histological part of the back of the eye. You see the photoreceptors in blue, and you see retinal pigment epithelium located right underneath photoreceptors. By the way, can you see my cursor as well? Can anyone confirm?
Yes, yes, I
Okay.
can see the cursor.
Thank you. So you see retinal pigment epithelium sandwiched between the photoreceptors, which are in blue here, and the choroidal blood supply. And this really location is strategic to how these cells function, because these are really the central part of this homeostatic unit in the back of the eye. These cells are highly polarized, and their polarization is determined by the tight junctions that are present between neighboring cells, which I'm highlighting here. And that really make sure that cells have special channels and proteins on the apical side and different channels and proteins on the basolateral side. And also, as you can see, anatomically or morphologically, these cells have different special apical processes that interact with photoreceptor outer segments on the apical side and sit on this proteinaceous Brooks membrane on the basal side for it to allow to interact with the choroid. And throughout their life, that's what these photoreceptors do. They take care of photoreceptors on the one side, perform several functions to help the photoreceptor survival and functioning, and also take care of the choroid in the back of the eye. So you can imagine something that is critical for both of these tissues. If something goes wrong with these cells, both of these tissues start to die, and that's what happens in this example of age-related macular degeneration, a disease that affects mostly elderly people worldwide. And what you see here in the bottom left corner is a stage of disease called dry AMD, where RP cells die off, and as a result of RP cell death, you see on top photoreceptors die and retina starts to degenerate, and on the other side, the capillaries start to die off, because these cells are making cytokines to maintain the capillaries, and they're helping photoreceptors with their metabolite and other chemicals. They are coming from the blood supply, oxygen and glucose that photoreceptors need for survival. Now, in another advanced stage of this disease of macular degeneration, something different happens, in which because of stress caused in RP cells, these cells likely secrete higher VEGF and other angiogenic molecules, which cause the choroidal vessels to overgrow. And in fact, they penetrate through what I call the Z direction. So instead of going in two planes, an XY plane, they go in a Z plane, penetrate through the RPE often, and leak fluid and blood into the retina, and again leading to blindness. So both of these conditions really end up with blindness in the dry form, blindness because of direct photoreceptor atrophy, and the wet form, indirect atrophy caused indirectly due to leak of blood and fluid in the retina. So our goal is to use really retinal pigment epithelium cells made from iPS cells to understand how these different stages of disease happen from initiation to progression, and to come up with a treatment for both of these stages. And I'll show you all of these various aspects throughout my talk. So first things first, we want to show when I really make RP cells from iPS cells, and we have optimized a very robust protocol to differentiate RP cells, mature RP cells from iPS cells. The process is about 10 weeks long, and it really requires a combination of different growth factors at different stages, which has been optimized based on previously published literature and a lot of work we have done in my lab over the past 5 to 6 years. And what we have also characterized is that the cells really go through these various developmental stages so that the epigenetic marks are correctly laid down as the cells differentiate into RP. They go through this neuroectoderm, RP progenitor, chromatic RP, all the way to mature RP stages. And in fact, we've recently published that we can really make functionally mature RP cells using our protocol. And at the bottom panel, you see we can also, the cells that we make are pure. Here's an example of a dish full of pigmented RP cells. And we can prove by flow analysis using several different RP markers we have done by now that we can easily get more than 98, 99% pure RPE cells. And something that is, if that's something that this protocol is so critical for us, for our lab, for most of the activities we do, we want to make sure that the protocol is reproducible, and that we have done by testing this over 30 different donors so far, for healthy donors, HLA matched iPS cell lines, clinical grade iPS cell lines, and several diseased iPS cell lines. So I'm not going to spend too much time on demonstrating that we can make functional RP cells. We have extensively published on it. As I said, just one recent paper and a previous paper in 2016, where we extensively characterize functioning of RP cells made from iPS cells, that these cells really behave like native RP cells with the protocols that we use for differentiation. They display, here's an example of electrical properties, their membrane resistance, transepithelial potential, and how they respond to various physiological stimuli, pretty much like how native RP cells respond to. So that I want to start with the first part of my talk to show you a little bit about 2D disease models and drug screening application of iPS-derived RPE cells. So in this case, in the first part of my talk, focus is going to be try to model the dry form of macular degeneration. We want to create a situation where RP cells die off as it happens in AMD. So to do that, we really dived into literature and looked for, what are the trigger for RP atrophy, what are the trigger for this disease. And it turns out that dry form of AMD is really initiated by metabolic insults and biological aging. And a number of reports published in the past 5-6 years have shown that if you treat in vitro RP cells in vitro with a complement competent human serum, you can trigger some of the phenotypes of AMD and hallmarks of aging. And this is aging quote unquote because we're doing it in a dish within a few days. But it really triggers all the hallmarks of aging and AMD in the cells. And so we thought maybe we can try to replicate those findings with iPS cell-derived RPE cells. And the idea would be that RP aging is triggered by this complement in the serum that causes inflammation in cells. So first we checked whether the aged cells, which are treated with the serum, does it show reduced functionality. In fact, when we treat these cells with such a serum, we can see here if we measure the transepithelial resistance of the cells, which is a measure of maturity, functional maturity of cells formed by these tight junctions between neighboring cell leading to electrical tightness of the monolayer. You can see control cells in a monolayer give you resistance of about 700 ohms per square centimeter, whereas aged cells within 48 to 72 hours dramatically drop their resistance to 250 or 300 ohms per square centimeter, suggesting that cells are not functioning as well as they're aging with this stressor. And one of the really hallmark of AMD is accumulation of ApoE positive deposits underneath RP cell called drusen deposit, which contain other oxidized proteins and lipids. And in fact, these cells treated with a stressor, which we call aged RP cells, have very severe accumulation of high accumulation of ApoE deposits on the basolateral or the underneath RPE cells. And not just ApoE, we have looked for other markers for drusen. They're there. Fibrillin 3 is another protein for drusen deposit. We also see high accumulation of lipid deposits. So really, hallmarks of AMD, as seen in patients' eyes, can be replicated in these cells. Drusen deposit are really shown by also by EM. So when we look at these cells by EM, on the left panel, you see control cells, nicely polarized apical processes, apically localized pigment granules. In fact, pigment migrating into apical processes, very typical of native RP cells, rarely seen in cultured RP cells, but we think because we mature our cells nicely, these cells also have pigment migration into apical processes. And when we age it, our RP cells, you can see apical processes are gone, cells look vacuoled, and underneath the cells, you can see deposits which are missing in control cells. And if I zoom up right here, you can see these really basal deposits that are reminiscent of drusen deposit seen in patient's eye. So with these, we're really confident that we have created a good model to study macular degeneration in these cells. And now we can start with patients' iPS cells and really try to correlate these phenotypes with patient genetics and uncover pathways that are linked to macular degeneration. Just to give you one example, we see that aged cells, as it's thought that aging is linked to higher inflammation, we see increased activation of NF-kappa B pathway as seen by cytoplasmic to nuclear translocation of P65 component of NF-kappa B in these cells. So with that, we thought these cells allow us the opportunity to excuse me, to discover drugs that can undo some of these hallmarks of aging or AMD in these cells. So with these phenotype, we performed a drug screen using a low-pack library which has about 1,200 pharmacologically active compounds, and looked for drugs that would protect cells from cell death caused by this stressor A23187, which causes proteotoxic stress, very similar to what happens in our cells. And the screen was performed for 48 hours. These red, blue, green, and yellow are 4 different 3D4-well plates, in which had different compounds. Each well had a different compound. And this, after primary screen and a secondary follow-up, and in which we identified 40 drugs, we performed a dose-response curve, 7-point dose-response curve of all those 40 drugs, and identified these 3 drugs that had reproducible effect of protecting RP cells from cell death in response to this stressor A23187 within 48 hours. This effect was seen reproducibly across more than one iPS-derived RP line. So that was the reason we picked these 3 drugs. And when we test these drugs on cells, sure enough, these drugs were able to undo activation of NF-kappa B, as shown by this example here. In aged cells, we see P65 in the nucleus, but when we treat these cells either with the blue, green, or red drug or yellow drug, we see P65 retranslates from nucleus to the cytoplasm, showing you that inflammation is downregulated in the cells, and we have preliminary evidence that these drugs are also able to undo some of the hallmarks features of AMD in these cells. So with that, I will conclude the first part of my talk. That I've shown you that we can generate functionally authentic iPS RPE cells. With these cells, we can replicate some of the early hallmarks of dry AMD, and we've done it from 5 independent donor iPS cell lines. We can perform 3D4-well drug screens using these cells very successfully, and with that drug screen, we have discovered a few drugs that we think can suppress disease-associated hallmarks in vitro, and that can provide potential drugs for patients going forward. So now I want to move to the second part of my talk, switch from 2D to 3D disease models. Because as I said in my introduction, the disease is complex, right? So it's not a disease of just one monolayer. It starts in the RP monolayer, but it spreads to the photoreceptors and the choroidal blood supply, both in the dry form where the 2 tissues die, or in the wet form where the choroidal blood vessels overgrow. So we want to be able to really create the disease at the 3D level and see what happens. How does disease actually spread from RP to these 2 cells to these 2 other tissues? What causes really RP capillary loss, or what causes capillary overgrowth? Is it just VEGF, or are there other molecules, other pathways involved in this? So to make a 3D model, now we need to add additional tissues to this model, in addition to RPE, additional cell types. And the next cell type we want to add here is endothelial cells. So just like we made RPE cells from iPS, we also make endothelial cells from iPS cells. This is a very simple protocol, much simpler than the RPE protocol, published by a group in 2014. It takes about a week and a half to get pure endothelial cells from iPS cells. Again, these cells go through the stages endothelial cells go through normally for differentiation. But we can easily get pure endothelial cells. And again, this process is reproducible across different healthy and patient iPS cell lines. So then we take these endothelial cells and we want to convert them into a capillary network. And to do that, we use a bioprinter, a 3D bioprinter. Many of you might be familiar with how this works, but this is essentially a printer that moves in a very precise XYZ orientation that can be controlled by a computer or by a program that you write. And there are these two needles, and as the needle heads move, they can print this what we call the bioink that contains endothelial cells, pericytes, fibroblasts, and hydrogel. And this needle moves, as you can see, in a very precise orientation and can print any pattern. And we make the zigzag pattern so that the cells would form a capillary network in this pattern. And as the pattern grows, as the capillaries grow, we can quantify how much angiogenesis has happened in the bioprinted tissue. So the way this works is we start with a scaffold made of PLG. I'll come back to the scaffold in the third part of my talk. We start with a scaffold. We flip it, and we print on one side with the bioink, like the way I just showed you, with, mixed with hydrogel mixed with cells. We flip it upside down, and we see the RP cells on top, and then we let it mature for 4 to 5 weeks, and the scaffold degrades, and RP and endothelial cells replace that space with an ECM that we call kind of a Brooks membrane equivalent. So it becomes really a 3-dimensional tissue that should really look like the back of the eye. And in fact, if you look at the tissue, that's how it looks. Really, RP cells on top and an area in between, which we call Brooks membrane, where the cells have secreted ECM, and it really contains most of the component of ECM component of Brooks membrane, and a capillary network in the back. And you can see the capillary network here marked in green and red is made more closer towards the scaffold, towards the RPE, and it's more dense towards the RPE, just like how it is in the choroid, and less dense further away from it. So if you zoom through the entire tissue, you can see there are capillaries of various sizes, and some actually span from the entire tissue. And this is stained in green for endothelial cells and in red for pericytes. And you can see the nice capillary network from top to bottom. This one is going all the way through tissue, and they have a lumen we think is about 20 to 40 micrometer in diameter. And RP cells nicely polarized on top of this 3D tissue. Here they are stained in green with ezrin, which is the marker for RP apical processes and also an indicator of RP polarization. And in red is marked for E-cadherin, which marks the RP tight junction. So it shows you RP cells nicely polarized on one side, and on the other side there's a nice capillary network, as you see with the red stain marked for PLVAP, which is a marker for capillary fenestration. So fenestration is a very interesting and important feature of choroidal capillaries. What it is that it's known that there's a kind of a small pore in the membrane of endothelial cells in the choroid. These pores are about 70 to 100 nanometer in diameter, and these allow for easy flow of macromolecules between the blood through the Brooks membrane into the RPE, and that's why these choroidal capillaries are called fenestrated. So while we do see high expression of fenestration marker in our printed capillaries, we wanted to make sure that these capillaries are indeed fenestrated. So we looked at them by TEM. As an example of a published capillary from human fetal RP, so from a cadaver eye, TEM, and you can see this is what the fenestration is. These arrows mark these pores in the membrane of endothelial cells. Now compare that to the pores that we see in our capillaries made with iPSC-derived RP and iPSC-derived endothelial cells, and I would say they're pretty similar. We get a very similar pattern of fenestration in the capillaries that we make in our 3D tissue. So we're very excited that we think we can also make tissue that is fenestrated and behave similar to the choroid of the eye. And also that we think that fenestration in this tissue is dependent on the presence of RPE cells. Because in this slide, what you see is we made this tissue in the presence of RPE or in the absence of RPE. And in the left, you see a 3D rendering of a small piece of tissue, which is about 100 by 100 micrometer in size. You see RPE cells in the background. And on top, you see high expression of fenestration marker and big and thick vessels. Whereas on the left side, on the right side, you see tissue without the RPE. And you can see the vessels are much thinner and they don't proliferate as much. And the expression of PLVAP is much weaker in this cell. So the expression of the fenestration marker is much weaker. And this was a very small piece, but if you look across the entire tissue, across the entire well, which is about 6 by 6 millimeter in size, you can easily see much weaker proliferation with the absence of RPE and much weaker expression of the fenestration marker. So we think both the capillary growth and the expression of fenestration is dependent on RPE cells. So this is exactly what happens in dry AMD. In the absence of RPE cells, capillaries die off, and it's also thought that at an earlier stage of AMD, the capillaries lose fenestration as RPE cells are not functioning properly. So we think we have created a model for dry AMD in a 3D model for dry AMD that has both RPE and choroid. So the next challenge for us was to create a model for wet AMD. And here's an example again of a wild type RPE and wild type endothelial cells 3D rendering of the tissue from top to bottom. You see RPE, and then you see endothelial cells vessels labeled in red, going up again, vessels, and then RPE cells. And then what we're going to do is we're going to do what we call a depth scan. So we're going to add pseudo colors to the tissue. And depth scan is you see anything that is at the choroid level is under the choroid is blue, choroid level is green, as you see most of the vessels are. Oops. As you see, most of the vessels are green at the choroid level. And if they're at the Brooks membrane level, they're yellow. And if they're at the RPE level, they are red. So you can see most of the vessels are green. Some are yellow, and there's no vessel that is red. Now, if I go to the next tissue in which we overexpress a transcription factor called STAT3 that we know causes inflammation in cells or increases expression of inflammatory cytokines, pro-angiogenic cytokines, including VEGF and other interleukins and interleukin-6 especially, what we see is that if you run this 3D rendering, you see RPE and then vessels, and then going back up, vessels and RPE. And now if you run the depth scan on this tissue, what you'll see is vessels that are green, but you'll see a lot more vessels that are yellow, so that are up here. And if you focus on this white circle, there's one vessel that goes all the way from green, yellow, and then it turns red. So that means there's one vessel that has gone completely in the Z direction from the choroid into the RPE, just like what happens in case of wet AMD. There's more vessel growth, proliferation in the sub-RPE zone, and there are some vessels that go through the RPE and leak fluid in the retina. And we see exactly something similar happening in our 3D tissue in a dish. So we think we have also created a 3D wet AMD model. So we're using both of these models to really further understand how disease progresses and what drugs we can use to kind of undo some of the hallmarks of AMD, at least in a dish. So to summarize this part of my talk, we have replicated early hallmarks of dry and wet AMD in a 3D disease model using patient-specific iPS-derived RPE and endothelial cells. We have structurally and functionally validated these models. I didn't have much time to show you all of that data. And now we're using this to discover some of the genetic pathways that lead to different stages of AMD in patients. Now, in the next few minutes, in the third part of my talk, I want to show you our approach to developing a cell therapy for AMD using patient-specific iPS cells. So the focus in this case is going to be of dry AMD. And what happens in this case is that because RPE cells die, photoreceptors start to die, and as a result, patients go blind. But if you look carefully in patients or in cadaver eyes, what you notice is between the central part of the eye where patients are blind, where photoreceptors are dead, if you go towards the healthy area, there's this area called the transition zone, where photoreceptors are not quite dead yet, but RP, as you can see, is gone. But these photoreceptors are destined to die. They're probably not functioning properly, and over the next several months of patient's life, these photoreceptors are going to die off, and the disease is going to keep progressing further. So the idea surgeons have that if you can bring with a patch of for RPE in the transition zone, you could protect the overlying photoreceptors from dying and stop the disease from progressing further. In fact, this has been surgically tried where people have moved a piece of RPE from the periphery of the same patient's eye into the center, and I've seen that in small number of cases where the RP cells, where that RP patch integrates into the back of the eye, patients' photoreceptors are spared, and the vision is stops from getting worse. And that is a very important proof of principle for us to really develop an autologous cell therapy, that if we can make a patch of RP from patients' iPS cells, patients on stem cells, then we could really make this stop this disease from progressing further and getting it worse. And that's what we're trying to do. There's a 4 by 2 millimeter RP patch on a PLGA scaffold, and usually cells on top and the scaffold in the back. And I'll show you all the preclinical studies that we have done, how this scaffold works, how to make this scaffold and to test the scaffold in animal models. So an autologous IND requires that a clinical grade RP patch is made at least from 3 patients, and it's tested both first using in vitro GLP studies, functionality of the patch to compare how the cells perform starting from different donors, and then to show with the GLP compliant fashion with preclinical animal studies, safety studies, to see that the cells and the patch is safe and doesn't cause any toxicity, and then to show efficacy in one of the animal models, either a rodent or a pig. So these are the studies that we have performed for our in preparation for our IND application, where we start with the clinical grade. We first optimized manufacturing of clinical grade iPS RP patch and made clinical grade RP patch from 3 clones, 3 iPSC clones per patient, developed the protocols and SOPs to make this patch. And then we tested the functionality of this patch from all 3 donors, and have tested now safety and toxicity at 4 different time points, cells from 2 different patients in almost 450 different rats. This study is completed, and have tested efficacy of this patch in a rodent model, and are now currently testing efficacy of this patch in a pig model. So all of this data together leads to an IND application that we are putting together. So I'll show you, in the interest of time, I won't be able to go into details of all of this, but I'll show you small highlights of each of these studies. So first, a protocol to manufacture a clinical grade patch. The process is long. It takes about 6 weeks, starting with draw of blood from patient day 0 all the way to manufacturing of patch. We go through a process of isolating blood, reprogramming the blood cells into a small bank, into iPS cells that we convert into a small bank of iPS cells at passage 10, which gets validated, and differentiation starts after passage 11. And we go on to making the cells, RP cells, similar, using the process I showed before as well, going through RP progenitor stage, seeding the immature cells at the scaffold, and making mature RP patch on the scaffold. Because the process is so long, we had to have, process go no go points, which we have put in place, starting with a bank of iPS cells which get characterized, and then during the differentiation process to check if the cells are differentiating well or if they're pure, and then finally confirming the sterility, identity, and purity of cells on the patch. And if the process fails, we have cryopreservation steps throughout the process that allow us to go back to one of these stages and manufacture the patch from this intermediate stage. So this is the scaffold that I showed you earlier too. It's a 400 nanometer diameter electrospun nanofibers of PLGA, and we make it such that RP cells can form a monolayer on top of the patch, which I show you here in the next picture. This is a scanning EM of RP cells grown on a patch. In blue, you see the patch. In brown, you see RP cells, and nuclei of the cells from the side, because you're looking at it from a side view, you see these apical processes of all the cells in green, and you can see then all the cells in the patch grow homogeneously, they differentiate homogeneously and have similar apical processes, and they're fully polarized. So then we went on to functionally characterizing this patch made from 3 different patients, 3 different AMD donors, and almost 3 clones per donor, performed using GMP grade protocols. Again, in the interest of time, I won't go into the details, but we checked at differences between patches from different donors, different clones at molecular level, morphological level, and functional level, and using all of these assays that I've listed on the side. This manuscript has been submitted, so hopefully it will be out soon. But the goal was here to, first of all, functionally validate the patch. Second, to see how much is a variation, technical variation from clone to clone within one patient. And third is to see how much is a variation from patient to patient. And finally, to identify some of the release criteria from some of these 6 assays that we're performing to come up with a final way to functionally validate the patch before transplantation as we manufacture it in GMP suites for patients. So all of this work has been done. We have picked some of these assays, especially transepithelial resistance and shape matrix, as potential candidates for release criteria. And I want to show you some of the data we have in animals to test this, where we have tested safety and efficacy of this patch. So first thing in animals, we're going to test whether the patch is safe. One of the issues people have, or a concern people have, is if any stem cells are left in your final product, that could lead to tumor, teratoma, or another tissue formation at the wrong place, and you wanna avoid that. So we, of course, demonstrate that iPS cells, there are no iPS cells left in our final product, and these iPS cells, and that iPS cells do not survive in our final product. But then to test it in animals, whether RP cells would actually, iPS-derived RP cells would actually lead to tumor or teratoma formation, we used immunosuppressed, immunocompromised rats. And this is an example in which we actually injected pure iPS cells in the subretinal space. And as expected, within a few weeks, these cells form a massive teratoma, as you can see in this slide here, with cells of various lineages in this teratoma that can be identified. But if we transplant an RPE patch made from iPS cells, we don't see, and this is, as I said, the study is done over 450 different rats. In not a single rat did we see any tumor, teratoma, or any other kind of tissue in the eye or anywhere else in the body. But interestingly, as the scaffold degrades within a few weeks, the whole patch, as you can see here in red stain for human marker, nicely integrates into the back of the rat eyes. So this tells you that if a patch transplantation is done, it will allow the entire monolayer to integrate and start functioning right away in the back of the eye. So, but to really test how things would look in an AMD patient, and to test the big piece, the 4x2 millimeter patch, we used pig as a model. The advantage with pigs is pig eye is very similar to human eye and in size and anatomy, so we could really test the tool, the surgery procedure we want to optimize for patients for transplantation of this patch. So, but the problem with pigs is that there's no genetic model that is similar to what happens in AMD patients. So to create that, we use laser ablation. So we use a micropulse laser ablation of RP. We use a micropulse laser of a wavelength that is absorbed only by RPE cells, and as a result, these cells heat up and they die off, and the photoreceptors would die, which you can see here at the bottom panel by infrared image of the eye is a laser damage RPE. You can see because RPE is gone, now the infrared can shine through the eye, so you can see the back of the eye. And in the right side, you see histology H&E stain, where you can see RPE cells are gone, some cells are clumped up, and as a result, photoreceptors are slowly dying. So the idea would be to transplant a human RPE patch here and protect the overlying photoreceptors from dying. So we developed a tool that can bring the patch to the back of the eye. The tool has a curvature that fits the back of the eye, it takes the transplant only in one orientation, and then it can be controlled by foot pedal, so the surgeon can maneuver easily the patch in and out and deliver it to the right place. So it's a surgery that shows you how the tool is delivered to the back of the eye. A pig eye has been prepped, and the surgeon is making the incision bigger to bring the tool inside. And in the next slide, the surgeon is going to deliver the tool, bring the tool here. The tool contains the patch. There is a cut in the retina. Retina has been lifted right here. The surgeon is aligning the tool tip to the cut of the retina and slowly will deliver the patch under the retina. And then the retina is flattened on top of the patch, and the patch stays right under the retina, which we can prove by non-invasive imaging of the eye by a technique called OCT, optical coherence tomography. You can see from the top view is a patch, and from the side view is a patch under the retina, shown here by yellow line, and retina looks fairly healthy on top of the patch. So now, to show that the patch is really functioning and is providing efficacy, we use a technique called multifocal ERG. So basically, we shine multiple light pulses to the back of the eye, and depending on where the light pulse hit, it gives us an electrical response from that part of the eye. So it tells us about the health of that part of the eye. For instance, if the light pulse hits the healthy part of the eye, it gives us this nice and robust big electrical peak. So it tells us that part of the eye is healthy. But if the light hits this laser injured part, which is right here next to the patch, and the patch is not there, you can see there's almost no response, suggesting that because of laser injury, photoreceptors are now dead and retina is dying. But if the light hits the transplanting part of the retina, where we put the human patch, you get a response that is in between, suggesting that the human RPE cells are able to protect the pig retina, pig photoreceptors to some extent, so that we can get some electrical response from that part of the eye. By histology, we can show that the human patch integrates even in the pig eye, just like the rat eye. You can see here human cells are labeled in green, and both pig and human retina, both pig and human RPE are labeled in red here. You can see pig RPE is labeled with this RP65 RPE maturity marker, nice and red label. And as it enters, this part enters to the human patch, which is also green, you can see these cells are starting to mature, and they're pretty much in direct continuation with the pig RPE. So they nicely integrate into the back of the eye. And they also protect photoreceptors, as can be confirmed by histology. If we compare these two stainings, this is from the same eye, area that was lasered but no transplant was given, versus area that was lasered but the transplant was delivered, which you can see right here. Compare the thickness of a photoreceptor nuclear layer in stained here by dark in blue. It's almost double the thickness of the area where there was no transplant, suggesting that human cells are able to protect photoreceptors from dying in the pig eye. So with that, I want to conclude and show you that we are towards the end of this project. We've been doing IND enabling studies for the past about 4 years. In green is everything that we have almost finished, which I showed you data on, tissue sourcing to generating GMP grade iPS cells, and performing validation of these cells and preclinical safety and efficacy studies. We're now in the process of writing our IND, and we hope to submit that soon. And I want to thank the entire team. And with that, I'll be happy to take any questions.
Great. Thank you so much, Dr. Bharti. This was a really great presentation, so thank you so much for taking the time today. So for those listening on the GoToWebinar, you can take the next couple minutes here to submit any questions that you have for Dr. Bharti. There's that little question box where you can submit those. While those questions are coming in, do you want to switch over to the other presentation, Marissa? Okay. So while those questions are coming in, I would like to mention to everyone that CDI is now offering human iPS-derived retinal pigment epithelial cells. So these cells are provided as a cryopreserved vial of cells. We have 2 different unit sizes, a smaller 1 million vial and a 5 million vial, as well as a 3-pack kit. So these cells are a very pure population of cells, at least 80% positive for BEST1, and they are, reproducibly produced, ensuring the same performance with every batch. So I just did want to mention that. We also have a program right now where we are awarding free vials of human iPS-derived neural cells. There's 6 different cell types actually that are part of that grant program, including the ISL retinal pigment epithelial cells. So this is out there to support innovative research, and if you'd like to enter, you can navigate to our website at cellulardynamics.com/grants. Just provide a brief description of your research aims and how CDI's cells would be used in your work, and winning applications will be awarded a free vial of cells to get your project started. Okay, so let's check in here and see what questions we have that have come in. Give me just 1 minute here while I pull that up. If we aren't able to get to your question in the next, looks like we have about a little over 10 minutes here left of the webinar. We will reach out to you, our support team will reach out to you directly with the email that you've provided so that we make sure that we get your questions answered. But hopefully we'll see if we can get to as much as we can here. Dr. Bharti, so have you examined any use of your approach for RPE damage associated with retinal detachment in addition to AMD? So retinal detachment causes general and extensive damage to the RPE.
No, we haven't. The simple answer is we haven't tested it any other condition. In fact, we have only tested it in a pig animal model that we showed for laser injury induced RPE damage. And we tested it in a rat animal model in which RPE is dysfunctional due to genetic mutation, and that leads to photoreceptor cell death. So there are only 2 cases where we have tested the efficacy of these cells, and they work in both cases. I don't see a reason why it would not work in that case. If it's done well in time, before when the retina is still healthy, it should work and should help reattachment of the retina in case of retinal detachment, but we haven't tested it yet.
Great, thank you. Another question here, more towards looking at the actual differentiation of RPEs from iPS cells. So we have someone interested in this protocol of being able to grow RPE cells from iPS cells in as little as 10 weeks. Previous methods took more like 6 months to create RPE clones from iPS cells. Has the process been accelerated, or are there different types of RPE-like cells that are made from iPS?
I would say the process has been accelerated. That's what I showed in one of the first few slides, where we show a schematic of how RPE cells are made. Because in the past it took 6 months because people used a process called spontaneous differentiation. So you let stem cells just randomly differentiate into any cell type, and including they would make RPE. But the process that we have optimized uses what is called directed differentiation, where we add specific growth factors at specific time points, guided by really all the animal studies that have been done to study development of the RPE. So really that causes cells to become RPE progenitors at a much earlier time point, and then eventually become RPE much faster.
Great. Okay. Another question about the scaffolds that are used for the transplanted RPE patches. So, do the RPE scaffolds form scar tissue, and when you transplant the RPE patches, do they form scar tissue?
So the one example I showed you of the rat, immunocompromised rat, in which we are able to keep the cells alive for a long time. As you can see, there's absolutely no scar when the scaffolds degrades, and in fact the whole monolayer nicely integrates. So we think if there's no immune response against the cells, there should be no scar tissue formed, and that's the advantage of using autologous cells, that there will be no immune response against the cells.
Okay. Any other, Simon, are you jotting down a question over there? No, you're good. I'm just waiting a little bit longer here to see if we have any other questions coming in.
Sure.
Sure. Okay. So, what is your opinion regarding using cells in suspension versus cells on a scaffold?
So, I think it depends on what stage and what kind of disease you are targeting. As I showed in my slides, that cells as a scaffold completely integrate. Whereas when they inject it as a suspension, the integration may not be as efficient. It will be there, but it may not be as efficient. So it really is a matter of efficiency of integration and how fast you want the cells to start functioning, because as they're transplanted in a monolayer, the whole monolayer we have tested is already functioning. So they should be kind of, so to speak, hit the ground running. Whereas if they're injected as a suspension, they will need time to integrate into the monolayer before they can start functioning. So it really depends on the stage and kind of disease you are targeting. The approach might be different.
Great, thank you. Okay, so we just have one other question here about modeling of AMD. So is there mathematical modeling of AMD due to metabolic insults? So for someone coming from a background of looking at metabolic networks, is there a mathematical model of AMD due to metabolic insults?
A mathematical model. That would be nice. We don't have one. No, that would be, I think that's an interesting thought. Well, but we don't have a mathematical model of AMD.
Okay. All right. Well, I think that's going to wrap up our Q&A session. I just want to thank you again, Dr. Bharti, for joining us today. Thank you to all of the members of the audience that have joined in. We will be providing a recording of this webinar. Is going to be available, and a link for that presentation is going to be sent out to the registrants today. And with that, have a wonderful Friday afternoon and weekend.
Thank you, everyone.